Good day, this is Professor Will, CBMD PhD. Today's Monday, January 6, 2020. It is 3:13:11 a.m. Eastern Standard Time. And this is a reading on microbial attributes of improved strains. So let's get started here. Microbial strain improved cannot be defined simply in terms of modifying the strain for overproduction of bioactive compounds. Strain improvement should also be viewed as making the fermentation process more effective. Some of the traits unique to fermentation process that make a strain improved are the ability to A. Assimilate inexpensive and complex raw material efficiently, B. Alter product ratios and eliminate impurities or byproducts problematic in downstream processing, C. Reduce demand on utilities during fermentation, air cooling, water or power draws, D. Excrete the product to facilitate product recovery, E. Provide cellular morphology in a form of suitable for product separation, F. Create tolerance to high product concentrations, G. Shorten fermentation times, and H. Overproduce natural products or bioactive molecules not synthesized naturally, for example, insulin. Need for strain improvement. Microbes, fungi, bacteria, actinomyces that live freely in soil or water and produce novel compounds of commercial interest when isolated from their natural surroundings are not ideal for industrial use. In general, wild strains cannot make the product of commercial interest at high enough yields to be economically viable. In nature, metabolism is carefully controlled to avoid wasteful expenditure of energy and the accumulation of intermediates and enzymes needed for their biosynthesis. The tight metabolic and genetic regulation and synthesis of biologically active compounds is ultimately controlled by the sequence of genes in the DNA that program the biological activity. To improve microbial strains, the sequence of these genes must be altered and manipulated. In essence, microbial strain improvement requires alteration and reprogramming of the DNA, or the genes in a desired fashion to shift or bypass the regulatory controls and checkpoints. Such DNA alterations enable the microbe to devote its metabolic machinery to producing the key biosynthetic enzymes and increasing product yields. In some cases, simple alteration in DNA can also lead to structural changes in a specific enzyme and increases its ability to bind to the substrate, enhances catabolic activity, or make itself less sensitive to the inhibitory effects of a metabolite. On the other hand, when the changes are made in the regulatory region of the gene, such as the promoter site, it can lead to deregulation of the gene expression and overproduction of the metabolite. A typical example is overproduction of the enzyme amylase, where specific constitutive mutants have been developed that produce the enzyme even in the absence of the starch inducer. Knowledge of the functions of enzymes, rate-limiting steps and pathways, environmental factors controlling synthesis further help in designing screening strategy. The outcome of the strain selection, however, depends primarily on the kind of improvement desired from the microbe. For instance, increased product yield that involves the activity of one or more genes, such as enzyme production, may be enhanced simply by increasing the gene dosage. Molecules such as secondary metabolites and antibiotics that are complex in structure and require coordinated as well as highly regulated biosynthetic process, however, may require a variety of alterations in genome to derive a high-yielding strain. Apart from modifying the strains genetically, the success of a strain improvement program also depends on developing and combining more efficient ways of screening, testing and confirming the improved and high-yielding status of the mutants against the background of non-improved strains. Improvement of industrial strains is clearly justified when one takes into account the additional anticipated capacity and extra fermenters capital cost required to meet the throughput in the absence of tighter genes and strain development efforts. Through strain improvement, one can free up fermenters and facilitate the launching of other fermentation products in the pipeline. Also of great importance is the use of genetically engineered microbes that manufacture non-microbial products such as insulin, interferon, human growth hormone, and viral vaccines that cannot be produced efficiently by other manufacturing processes. Significance, impact, and benefits. Strain improvement is the cornerstone of any commercial fermentation process. In most cases, it determines the overall economics. Fermentation economics is predominantly determined by manufacturing cost per unit of product made, dollars per pound, and the costs associated with plant construction and startup. Although lower fermentation manufacturing and capital costs can be anticipated from fermenter engineering, design improvement, and microbial strains, offers the greatest opportunity for cost reduction. Great efforts are therefore expended to develop industrial strains that have an increased ability to produce the compound of interest at a faster rate. Great efforts are therefore expended to develop industrial strains that have an increased ability to produce the compound of interest at a faster rate. Enhance productivity of the fermentation process through strain improvement. More product. Vessel unit time, e.g. grams per liter per hour, is one factor that makes the most impact. It can determine the ability of manufacturing process to meet additional demands without adding more fermenters. Furthermore, the strain that can synthesize a higher proportion of the product using the same amount of raw material can also reduce material and manufacturing costs significantly. Strategies for strain development. Several procedures are employed to improve microbial strains, all being about changes in DNA sequence. These changes are achieved by mutation, genetic recombination, and the modern DNA splicing techniques of genetic engineering. Each procedure has distinct advantages. In some cases, a combination of one or more techniques is employed to attain maximum strain improvement. Mutation. Microbes generation after generation generally inherit identical characteristics as their parents. However, when changes occur in the DNA, they too are passed on to daughter cells and inherited in the future generations. This permanent alteration of one or more nucleotides at a specific site along the DNA strand is called a genetic mutation. The strain that harbors the mutation is called a mutant strain. Although a gene consists of hundreds or even thousands of base pairs, a change in just one of these bases can have a significant change in function, operation, expression of the gene, or in the function of its protein product. The mutant, by definition, will differ from its parent strain in the genotype, a precise nucleotide sequence in the DNA of the genome. In addition, the visible property of the mutant, its phenotype, may also be altered relative to the parent strain. A point mutation may be associated with a change in a single nucleotide through substitution of one purine for another purine, or substitution of one pyrimidine for another pyrimidine transition, or through substitution of one purine by a pyrimidine, or vice versa transversion. Mutations may also result from deletion of one or more base pairs, insertion of base pairs, or rearrangement of the chromosome due to breakage and faulty reunion of the DNA. These changes in base pair arrangements can alter the reading frame of a gene frame shift. Mutations, and during its transcription and translation process, also changes the amino acid sequence of the resulting protein. Most mutations occur on the chromosome structure at a specific site or locus gene mutation. Genetic mutations do occur spontaneously at low frequency at any point along the gene, 10 to the minus 5, negative 10 to the minus 10 per generation. Some mutations are the result of integration or incision of insertion sequence elements and result in subtle modification of genetic sequence. Potential for a microbe to mutate is an important property of DNA since it creates new variation in the gene pool. Modification of the strain through mutation can also be induced at will by subjecting the genetic material to reaction with a variety of physical and chemical agents called mutagens. Most cause some damage to the DNA through deletion, addition, transversion, or substitution of bases or breakage of DNA strands. Although microbes have systems to repair the damage or alter DNA and return it to the original form, the repairing and editing mechanisms are not error-proof. Thus, when DNA reacts with mutagenic agents for longer periods of time, the damage in the DNA cannot be repaired to the correct genetic sequence with the same rapidity and accuracy in the normal circumstances. The microbe's progeny that survives the changes in their genetic DNA sequence usually require an altered genetic code for reprogrammed metabolic and biosynthetic activity. The frequency of bacterial mutation for a particular trait is low, 1 per 10 to the power of 4 to 10 to the power of 10 cells per generation. In addition to mutation, alteration in DNA can also occur by genetic recombination. Here, information from two similar but different DNA molecules is brought together and recombined by crossing over, resulting in novel DNAs of different lengths so that new combinations of mutations are produced. This allows the circumvention of slow leaks to obtain new combinations of desired characteristics in microbes. Genetic engineering is usually employed to create targeted mutations on the genes, unlike other methods of mutation that are random. It must be emphasized that this technology is not a way of constructing new forms of life. Even the genetic materials of the simplest organisms are highly complex, and insertion of a few genes from an unrelated organism will not create a new microbe. One of the basis of the method of screening and selection chosen. There are basically two methods of improving microbial strain through random mutation. Random selection and rationalized selection. Random selection, random mutagenesis and selection is referred to a classic approach to non-recombinant strain improvement procedure. Improved mutants are normally identified by screening a large population of mutated organisms, since the mutant phenotype may not be easy to recognize against a large background. After inducing mutations in the culture, the survivors from the population are randomly picked and tested for their ability to produce a metabolite of interest. Moreover, it offers a significant advantage over the genetic engineering route alone by yielding gains with minimal startup and sustaining such gains over years despite a lack of scientific knowledge of the biosynthetic pathway, physiology, or genetics of the producing microbe. This empirical approach has been widely adapted by fermentation industry, following the successful improvement in penicillin titers since World War II. One drawback to the random selection approach is that it relies on non-targeted, non-specific gene mutations. So many strains need to be screened to isolate the improved mutant in the mixed population. In addition, when the culture is magnetized, multiple mutations may be introduced in the strain. This may result in the enfeeblement of the organism lacking the properties of interest. This process of strain improvement involves repeated application of three basic principles: mutagenesis, the population of induced genetic variability; 2. Random selection and screening from surviving populations of improved strains by small-scale model fermentation; and 3. Assaying of fermentation broth agar from products and scoring for improved strains. It must be emphasized that the action of the mutagenic agent on DNA can not only cause genetic alteration but can also induce cell death, owing to irreversible damage to the DNA formation of lethal mutations. After the mutagenic treatment, mutants are sought among the surviving population with the anticipation that each of the surviving cells harbors one or several mutations. Each time an improved strain is derived through mutation, it is used again as the parent strain in a new cycle of mutation screening by fermentation in liquid or solid and assay. This random procedure of mutant selection is continued until a strain is derived that is statistically superior in performance compared to the control strain prior to the mutagenic treatment. The objective of mutagenesis is to maximize the frequency of desired mutations in a population while minimizing the lethality of the treatment. For this purpose, nitroguanidine (NTG) has been a mutagen of choice because it offers the highest possible frequency of mutants per survivor. The efficiency of the random selection process is dependent on several factors: the type of culture used, such as spores or conidia, mutagen dose of exposure time, the type of damage to DNA, conditions of treatment, and post-treatment frequency of mutagen treatment, and the extent of yield increase detectable. In addition to the mutation, conditions to test are quantitative and analytical screening procedures employed, bioassays, radioimmunoassays, chromatography, HPLC also play a critical role in successful isolation of superior mutant. The ability to detect the gained mutant among the randomly selected mutants is greatly influenced by the process, as well as the variability within the process and the actual titer differences between the improved strain and the control. The screening procedure, therefore, is usually designed to maximize the precision and selectivity of improved cultures gained per sample tested, and to minimize the variability measured as the coefficient of variation when treating the unmagnetized control and reference samples. All the strains tested, including the control, are normally worked up all the way head-to-head from the initial cell clone stage to the final screening stage. This is essentially the test of significance. As such, if all the treatment conditions were the same, as successful tests should show a statistical difference between the means of the control and improved culture. Good day, this is Professor Will, CBMD PhD. Today is Monday, January 6, 2020, 5:34, 12 seconds a.m. Eastern Standard Time. This is the end of my microbial strain reading. Hope you enjoyed listening. Have a great day.